All right. Well, I'd like to welcome everyone to today's Polyploidy webinar here on February 5th, 2025, our first webinar of the year. And today we have two speakers, and we're sort of going in reverse order from what we expected to get around some Zoom screen sharing issues. Our first speaker today is Christoph Rosche at the University of Halle. Christoph's research focuses on disentangling population ecological processes that drive the local spread or decline of plant populations. He's particularly interested in how environmental gradients determine the genetic structure of populations and the phenotypes among population variation through local adaptation. He also uses biological invasions as models to study eco-evolutionary principles at contemporary time scales. Today, Christoph's going to tell us about recent work on herbarium specimens reveal a cryptic invasion of polyploid Centaurea stoebe in Europe. With that, I'll let Christoph take it away.
Thank you so much, Mike. This was really nice and perfectly introduced my talk today. So thank you again for the invitation and also for the nice introduction. So herbarium specimen reveal a cryptic invasion of polyploid Centaurea stoebe in Europe. And all right, so let me start with a fundamental opinion paper of that many of you probably are familiar with. So that's Van Drunen and Johnson's view on polyploidy in urban environments. And in a simplified way, they describe how, can you see my mouse pointer? Do you see the pointer? Yeah,
yes,
perfect.
looks good.
In a simplified way, they describe how the occurrence of polyploids increases along an urbanization gradient, meaning that the more urbanized an area is, the more frequently polyploids appear compared to diploids. So for those who haven't read the paper yet, I highly recommend it. They included this fun cartoon and that illustrate different mechanisms in a pretty cute way, along with hypotheses and how they could be tested. But I won't go into that now. I would rather stick to my main takeaway. Polyploids appear to be more successful in rural habitats than diploids. And this idea pretty much aligns with a classical paper by Tebes et al. It's a review, and they argued that polyploid species are overrepresented among invasive species. So in their review, they illustrated this pattern, for example, here using data from 5 selected plant species with polyploids shown in black and diploids in white. Tebes went through various case studies and focused particularly on Centaurea stoebe, so the spotted knapweed in Asteraceae, which is native to Eurasia and invasive to North America. And just some context for this invasion, the spotted knapweed is among the most devastating weeds in North America. In this photo here, you can see that it builds up virtual monocultures, for example, here on a ranch in Montana. And the species covers an area of 3 million hectares, which equals to the size of Belgium. So if you put it like all together in North America, it has the size of Belgium, which is quite a lot for a European at least. And it causes $150 million damage per year, mainly by making rangeland unusable. So, but the most interesting part, Centaurea stoebe is a polyploid complex of a diploid and a tetraploid cytotype, and both cytotypes occur in the native range. Here you see, where diploids actually represent the majority cytotype, and in strong contrast, only tetraploids were able to colonize the novel range. And this striking cytotype shift results in 3 so-called geocytotypes, and Tebes et al. Called it an excellent model for evaluating the role of polyploidy in plant invasions. Some more background about the model. Diploids and tetraploids are pretty much similar in the morphology and in the ecology, so they both can occupy basically the same habitat types. And the most important difference is that tetraploids are polycarpic perennials, whereas diploids are predominantly monocarpic. tetraploids also slightly prevail in disturbed habitats like roadsides, whereas diploids tend to occupy more natural habitats such as dry grasslands, and tetraploids appear to be more resistant to disturbance in clipping experiments. Okay, so far so good, but there's one particular problem. There's increasing evidence that many successful invaders do not only spread in terms of classical transcontinental invasions, but also simultaneously expand their native range. And a recent synthesis by Zhang et al. Showed that in Europe, for example, the number of naturalized non-native plants with intracontinental origin exceeds those with transcontinental origin. And yeah, and our understanding of these gradual expansions of native ranges is quite limited because the precise border of the, let's say, true native ranges of species are usually unknown or speculative. And this lack of information may be particularly common in polyploid complexes, so where cytotypes are difficult to distinguish morphologically, like in Centaurea stoebe. So this means range expansion of one cytotype can go unnoticed into areas where only the other cytotype is already present. And these expansions can be regarded as cryptic invasions. And a review by Moraes and Reichert showed that papers on cryptic invasions have been increasing, but the overall number of these studies is quite low. And additionally, most of these publications focus on aquatic habitats, while terrestrial habitats have received much less attention. And this is exactly where our research comes in. So the following slides will present results from a current paper, which we just published in New Phytologist a month ago. And in this study, we investigated whether Centaurea stoebe has expanded into European areas where it was not originally native. And if so, did both cytotypes expand equally, or have there been a cytotype shift in Europe? And more specifically, the question is the cytotype frequency shift, so the invasion of North America by only tetraploids, is this solely a transcontinental phenomenon, or are there similar biogeographical patterns within the sympatric European range or Eurasian range? So what we did, we revised more than 13,000 European herbarium specimens of diploid and tetraploids from 167 herbaria. So the purple dots here are the herbaria, and the size indicates the number of specimens investigated. And this was quite tedious work because the two cytotypes are really hard to distinguish. And this huge amount of work was done by this guy. The senior author of the paper, Patrick Marzi, is an incredible taxonomist. And here you see two specimens where the key distinguishing traits are most pronounced. In diploids here on the left, you see that these capitula, they tend to be rounder compared to tetraploids here on the right, number 2. And additionally, tetraploids often have accessory rosettes at the root collar, which you rarely can find in diploids. Yeah? And this accessory structure is linked to the polycarpic life cycle of tetraploids. Okay, after the cytotype determination, what we did next was georeferencing of the specimen. So we assigned each specimen to an exact location, and whenever possible, also to a distinct habitat type. And we observed that Centaurea is nowadays found mostly in rural habitats, such as roadsides, railways, and other highly disturbed areas. But to understand where the native distribution of the species is, we ask, in what kind of habitats did the species naturally occur before humans completely changed the world that we are living in? And the answer is, so this is a light-demanding species, and it originally occurred in two main habitat types. First, steppe-like habitats, so dry continental grasslands, like here in a photo in the Ukraine. And the second is so-called relict habitats. So this can be steep stony slopes, often found in high mountains, as here in the Alps, and rocky outcrops in low mountains or along river canyons. And the occurrence of tetraploid populations in these relict habitats were particularly interesting to us because Centaurea stoebe is a predominantly burrow species with a limited uphill dispersal. This colonization of these relict sites is very unlikely without the presence of nearby source population over an extended time period. And we used the spatial patterns of these habitat-specific distribution information plus phylogeographic information that we took from several population genetic data sets to distinguish between native and expanded ranges of tetraploids and diploids. I won't go further into details for this, as I don't want to give a methods talk today, but here you can see the outcome. So this is a map of both cytotypes in Europe. So for diploids, the blue points, we found that wherever they occur in Europe, they consistently can be found in steppe or relict habitats. And in contrast, tetraploids are only native to this part. This native range includes mountains in the Balkans and Romania, plus some natural steppes until West Russia. And they expanded their range towards Central, North, and Western Europe. And this cryptic invasion, which was unnoticed, resulted in a wide overlap of the distribution of both species, of both cytotypes. We made this assessment without considering a temporal component, meaning that we didn't use the collection dates to define the native and expanded ranges. So only posterior, we look at how the two cytotypes have occurred across Europe across 50 years in the walls. So on the left, you can see the distribution of diploids until 1872, and then followed by the next 50 years, and then following two 50-year periods. And what we see is that diploids have a very stable occurrence pattern regardless of the time period. So their distribution appears consistent over time. Yeah. In contrast, tetraploids, up to 1872, there were hardly any occurrences outside the native range, except for a few records in the immediate vicinity, such as Budapest, which is a major trade hub in Europe at that time. And in the following time periods, the distribution of tetraploids expanded significantly towards Central Europe and Western Europe. So next we ran GAMS to estimate the relative proportion of tetraploids to diploids over time separately for the native range in orange and the expanded range in red. And this thin horizontal line indicates a proportion of 50%. In other words, if you randomly choose a specimen from a big box with particular distinct spatiotemporal information, you would have a chance here of 50-50 to get either cytotype. So in the native range, there was no significant change over time. We've seen in the dashed line, so the proportions of tetraploids remain stable at around 50%. Sorry. But in the non-native range, in the expanded range, we see significant temporal changes. Tetraploids started at 0 with a slow spread between 1850 and around 1920. And after that, the frequency of tetraploids remained stable for a while, and then again increased exponentially, and now just reaching over 50% in the expanded ranges. To investigate what influences how long tetraploids have been present at a given location, we performed boosted regression trees. For this, we transformed our dataset into 10-kilometer grid cells and analyzed how various characteristics for these grid cells here on the y-axis affected the year since when tetraploids were first recorded. So the so-called residence time here on the x-axis. And here you see the relative contribution of different predictor variables as individual bar plots, or summed up to 4 different categories in the pie chart, including spatial data in dark green, climatic data in black, anthropogenic factors related to dispersal in olive, and to urbanization in orange. Without going too much into the detail, the factor space was obviously the most important one for the initial spread. I will now highlight the 3 most important variables. So the most important variable was the distance to the native range. The further a grid cell was away from the native range, the shorter the residence time of tetraploids. In other words, tetraploids are longer present in areas that are close to the native range. Yeah? And we found similar patterns for the latitude, whereas the connectivity index showed a positive relationship. So tetraploids are longer present in areas that are well connected in terms of traveling and trading infrastructures. We also analyzed how ruderal, gray, and natural habitat types green differ between the cytotypes for diploids and tetraploids. So on the left, you can see the distribution for diploids, on the right for tetraploids. And for diploids, we observe similar distributions between Central Europe and Southeastern Europe. In contrast, for tetraploids, we see a clear spatial pattern. Natural habitats are far more common in the native than in the expanded range. An important detail in this figure is that these natural habitats are not necessarily the habitats that we use to define the native range. Remember, these habitats were natural steppes and relict habitats, but these only account for about 10% of the natural habitats, what are shown here, because these also include semi-natural habitats, for example, grazed grasslands. So that's why we also see some green points in the expanded range, much more for diploids. Yeah? And we then analyzed this pattern using another GAM, specifically focusing on tetraploids within their expanded range over time, so only in the expanded range here, distinguishing between ruderal and natural habitats. And there is a slight increase in occurrences within the natural habitats, but the trend was much stronger pronounced in ruderal habitats. So look, the frequency of tetraploids in the expanded range is nowadays way above 50%. And we then zoomed into this part of the curve. So the exponential increase approximately coinciding with the onset of the Anthropocene. And we were running another GAM only using data from the expanded range ruderal habitats since 1945. And this is the curve we got. And we contextualized the temporal patterns with political changes in Europe that arguably influenced travel and trade between the native and expanded ranges. And we found some interesting correlations. No proved causation whatsoever, but there are some interesting patterns, such as this particular increase corresponding with the fall of the Iron Curtain, which obviously increased trade and travel between native and expanded ranges. So we took this time period since 1989 to take a closer look at the current spread of tetraploids and identify the key factors influencing this current expansion. We ran another boosted regression tree using the same variables on the x-axis, but on the y-axis, the proportion of tetraploids since 1989 on the x-axis. And in this model, spatial factors here in green were no longer the main drivers, but instead variables related to urbanization became more important. Yeah. On the next slide, I will take again a closer look to the 3 most important variables influencing this current spread. Here you see the current spread is strongly linked to impervious cover. So the more concrete per grid cell, so the more asphalted an area is, the more frequent we find tetraploids. Additionally, the greater the row density within a grid cell, more tetraploids can be found, and then latitude has a slightly negative effect, meaning the tetraploids become less common further north. All right. So the key question is, what do we actually learn from the study beyond looking at nice figures? First, cryptic invasions. Moraes and Reichert, they already suggested that cryptic invasions are likely much more frequent and ecologically significant than we assume nowadays. So our study strongly supports this assumption, and to better understand how widespread this phenomenon actually is among terrestrial plants, we simply need more case studies on spatio-temporally explicit distributions of the polyploid complexes. Second, the issue with the true native ranges. So tetraploids are only native to southeastern Europe and expanded toward Central Europe, and similar range expansions have been proposed for various plant species. This pattern is possibly influenced by both climate change and pronounced human activities in Central Europe. However, we lack quantitative assessments on spatial temporal range shifts of species, and this hampers our ability to identify native range expansion, also the drivers behind these expansions. Yeah, and our study demonstrates that herbarium specimens actually can provide invaluable resources for so far lacking data on spatial temporal range dynamics of polyploid complexes. The third, with respect to our study species, a particularly interesting scientific problem emerged. So far, there have been 53 publications that examine differences between the geocytotypes, yeah, but none of these studies has distinguished between native and expanded populations when they took European populations. So let me show you this table here. What you can see is an overview of some of these publications, and the first column is for the reference, the second one is the comparison they made, so either between ranges or between cytotypes, and the third column is the number of citations of each study, and most importantly, the last column shows the percentage of European populations used in this study that are based on our assessment actually not native. And this represents a huge body of research with over 5,000 citations in total. And what stands out is that on average, 77% of the European populations that had been used in these studies are likely not native. And this raises important questions about how solid are the findings in the existing literature, including my PhD studies, by the way. And yeah, we therefore strongly recommend a careful revision of spatial temporal range dynamics of your model species before conducting your sampling, especially if you are working with polyploid complexes. All right, and the fourth point, our study supports the opinion that polyploid species are better at colonizing non-native ranges and urban environments, and the strong colonization ability of tetraploids in roadsides and habitats with high impervious cover seems to play the key role for the success in tetraploid Centaurea stoebe. So, given the ongoing fragmentation of natural habitats, increments of rural habitats, this distribution may indicate that polyploid relative to diploid plant species may benefit from increasing human activity under global change. So I believe it's crucial to conduct more studies in this direction to test generalizability of our findings. This is important because the trends described in these two reviews have also faced major criticisms, with some arguing that they are not universally applicable, but rather subject to specific taxa or other limitations. And we are currently preparing first steps towards a new review that aims to re-evaluate whether polyploids are really more likely to become invasive than diploids. I believe the best were right there, but it's still a long way to go before we can make any clear conclusions. And if you have thoughts on this topic, please reach out to me. I would love to discuss this further. And finally, the last slide, I would like to briefly mention our ongoing studies. So we are currently analyzing North American herbarium specimens, reconstructing spatial-temporal expansion routes, which also includes tracking climate niche shifts as illustrated here, so from green to dark red, indicating a niche shift in comparison to the native range. And we are now measuring plant traits such as capitular size directly on herbarium specimens across both continents, Europe and North America. And I'm hoping that the results will provide insights in why and how tetraploids spread in certain spatial, temporal, and climatic contexts. And if anyone is interested in this new research or even wants to collaborate, I would also love to connect, because this is currently my absolutely favorite project. Okay, for now, please just let me thank you. Thank you so much for your attention.
All right. Well, thank you, Christoph. That was really wonderful. We have a couple of minutes here before we transition back over to Charity. If there are any questions, please feel free to put your hand up in the Zoom box or pop a question in the chat. All right, Justin Ramsey, please go ahead.
Hello, hopefully you can hear me. Great talk, really enjoyed that.
Thank you.
Question, do you think that the traits of the tetraploid, so the size and shape of the capitula and the polycarpy, does that make sense based on them being tetraploid, or is it just sort of a coincidence that the tetraploids have those traits?
In terms of the size of the capitula or the polycarpy? So the size of the capitula I'm currently measuring because I just want to have some proxies for how they perform over time and space and so on. And I do think that the polycarpy actually, so the longevity, the increased longevity, is a big thing of colonizing. There are reasons to overdue or to overcome issues of founder populations such as inbreeding depression and limited mates and all these kind of things. Disturbance tolerance is much better due to this polycarpic life. If you have like a strong disturbance, then you kill basically all diploid individuals, and the tetraploids just can resprout. I think that's the major thing in case of Centaurea stoebe, and I think that especially for these allopolyploid mountain-like species where we have a lot of these polyploids, there are many that evolved polycarpy or at least increased longevity, and that could be a major thing shaping biogeographical patterns. Yeah.
And do you think it's the increased DNA content, the nucleotypic effect, that gives the plants those traits, or is it something else?
That is a good question, probably better related to people with a more mechanistic background in polyploidization, such as Douglas, who was just raising the hand. But I think so, yeah, I think so.
Thank you.
Thank you, Justin.
All right, well, Doug, go ahead and fire away.
Hey, thanks, Mark, and thanks for that was a great talk, really fascinating. Yeah, so a couple of things, and probably some of these are best answered with genetic tools, but we talk about the polyploid, in this case tetraploid, moving into these rural habitats, but is it just some populations that have made the jump? I mean, some genotypes of the tetraploid, or are they all able to do it? And I guess the other question that's kind of related to that, are these tetraploid plants that are now adapted to these, let's just call it new invaded space, are they already genetically distinct or have slightly different adaptations than the native plants? That is, if you grew like an ecological habit, where you just had these plots, are they even the same anymore? Have they already differentiated? Is it not just a cryptic invasion, but a cryptic differentiation?
Yeah, that is a good question. And there's this Hufbauer. I think Hufbauer had this anthropogenic induced ability to invade theory. I also found something. There was this study, I don't have it here So we did this clipping to demonstrate how they are tolerant against ruderal stress. And we did find differences within the cytotypes regarding whether they are ruderal or natural populations. Yeah. When it comes, unfortunately, I don't have this graph here. When it comes to genetics, in my PhD, we did microsatellite analysis for both European range and North American range, and for tetraploids, it was a mess, a total mess. So we couldn't find any genetic structure, neither in the non-native nor in the native range. A bit more of structure in what we now assume is the ancestral original range, but in general, there was hardly any structure, whereas in diploids, you had a clear, let's say it was more of a latitudinal gradient of k equals 2 clusters. So I wouldn't say that only particular genotypes or even a specific lineage involved this expansion, at least not from the messy microsatellite data that I had. Yeah.
All right. Well, thank you. And we got one very quick question, maybe while we switch slides, if Charity can, Christoph, if you could stop sharing, Charity can try to get her sharing. And then in the meantime, we can let Andre's question here that's in the chat.
Do I have to do something for this?
You should be able to stop sharing, is what you should do.
Okay. Yeah, I'm going to try.
Perfect.
Oops. Where am I? You just have it. Here, there's the question. Thank you for such great talk. Very nice.
So how do you define the native range for polyploids in Europe?
Yeah. Yep. I would like to stop the sharing in the meantime. Here, Freigabe stoppen. Now I got it. Now I should be fine, right?
Good. Yep, it looks good. We see Charity. Perfect.
Perfect. So the last question. How do you find the native range for polyploids in Europe? So basically, the main thing is assuming that we go like 5,000 years ago back in time, everything what we have in Central Europe should have been more or less woody area with some rocky outcrops where such species had relict habitats, where they were still present from glacial times when we had like cold steppes in Europe. And diploids, for example, they just went on these rocky outcrops when everything was wooded again, and we can still find them there, and we don't see tetraploids over there. So they just stayed where they are around the Black Sea in these steppes and in some mountains in the Balkans. So that was the main definition. It was a very long process, just short in a nutshell.
All right. Well, I think we'll get things started up. There we go. Looks like Charity got it sharing again. All right.
I think it kicked me off after a minute, but hopefully
I don't
it
know
doesn't
what
do
that
that
was
again.
about.
I don't know either. All right.
We only see your background there, Charity. So I'll let you
no,
get
that's okay. I haven't hit share yet.
Oh, perfect.
Now you can see it, right?
Yeah, excellent. All right.
Thank God.
Well, let me introduce you for just a second. So
Okay.
So Charity Goeckeritz is a NSF postdoctoral fellow working at the Harcus lab at HudsonAlpha, where she's studying apomixis and the Rosaceae. And you may remember Charity; she was on here 2 or 3 years ago. I can't remember now, telling us all about her work during her PhD on sour cherry genomes. And today she's here to talk to us about genomics of triploid apples, narrowing the search for the genetic basis of apomixis in Malus. With that, I'll let Charity take it away. Thank you.
Great. Thanks, Mike, so much for organizing. And I really appreciate Christoph, that very nice talk, and for going first while I fix these technical difficulties. So yes, my name is Charity, and I'm an NSF postdoc in the Harcus lab at HudsonAlpha, and I study this very weird phenomenon called apomixis in Rosaceae. So today I'm going to be sharing some of the results I have for the apple side of my project. I also work on blackberry, but I won't be really touching on those guys today. Oh God, am I going to have to click? There we go. Okay, so first things first, I always like to take a moment to orient myself and others in relation to the structures and the developmental processes we're talking about when we're talking about apomixis. So we're going to start pretty broad with the tree. And on that tree, we have flowers. And deep inside the flower ovary is this weird alien looking structure, which we call the embryo, or which we call the ovule, and that houses the embryo sac. So the most important cells in the embryo sac for plant reproduction are the egg cell and the central cells. Both need to receive sperm from pollen in order to form a healthy seed if we're talking about a sexually reproducing plant. But for apomictic plants, which can reproduce clonally via their seed, there's a few key developmental differences that occur. So the first is that the cells of the apomictic embryo sac are clonal because during embryo sac formation, meiosis is skipped. It's replaced with mitotic divisions. And we call this process apomiosis. And the second major difference between apomictic and sexual reproduction is the ability of the clonal egg cell to spontaneously begin forming an embryo, which we call parthenogenesis. So it does not need to fuse with a sperm nucleus through the process of pollination. And we now know that these two components of apomixis are controlled by independent genes. So in most cases, though, both reproductive modes need fertilization of those central cells to produce the nutrient reserve for the developing embryo, which we call the endosperm. And some other terms that we have to go through too are we have what we call pseudogamous apomix, and those require pollination of the central cells for that endosperm formation. But we also have autonomous apomix, which do not. And finally, we have to address the fact that almost all apomix are facultative, and that means they can reproduce sexually or asexually. So this includes apple. And how often an apomict does one or the other is referred to as the penetrance of the trait, and it's kind of a hot topic in the field that we honestly don't understand very well. So most Rosacea apomix, including apple, are facultative, and they exhibit a type of apomixis called apospory. So if you're curious as to what this process might actually look like in apple, here are some confocal images of a species that is apomictic called Malus sargentii, and the top row shows the developmental stage of the ovule, the bottom row showing what the flower looks like externally at that point. So at some point during ovule development, the megaspore mother cell emerges. It undergoes meiosis to form a linear tetrad, but in this species, this is kind of where the sexual process stops. And the surviving megaspores should have underwent mitotic divisions here to give rise to the sexual embryo sac. But instead, you see a somatic cell kind of appear out of nowhere, and we call that the aposporus initial, and it assumes megaspore-like properties, jumps right into embryo sac formation, and finally, you end up with an aposporus embryo sac that contains cells and are clonal to the mother plant. So I study apomixis for a few reasons, one of these being its importance in plant breeding. I'm a hardcore geneticist, and I'm kind of interested in genetics and polyploidy evolution together, but I'm like the functional genomics gal. So one way apomixis could revolutionize plant breeding is by preserving hybrid vigor, which is of course a lot of what plant breeding is built on. And it's the phenomenon where you can cross two parents and get progeny that are like way better than both parents, sometimes far beyond our expectations. But the problem with that is that if your plant is sexually reproducing, you have to keep maintaining those parent lines virtually forever if you want to recreate that really great progeny. And you can't save the seed, even if it happens to be self-fertile, because meiosis occurs and shuffles that DNA in both of the gametes. But if you have an asexual lineage, you can save the seed and it'd be the same individual. So that's kind of where this is really a great phenomenon to study for plant breeding and improving that efficiency. So thanks to decades of research, we do know a little bit about the genetic basis of apomixis in a few different plants, but this is my connection here. The first important thing to know is that the vast majority of apomix are polyploids, and the genes controlling apomixis typically have dominant effects. So 2 apomictic genes in 2 species have been well characterized through classic genetic mapping studies. And in grasses, the genes controlling apomixis are in one large hemizygous region, and they're linked. So the components for parthenogenesis and apomiosis are in the same region, and recombination is really heavily suppressed there, probably due to this large amount of hemizygous sequence. But in dandelions, it's actually not the case. The genes for apomixis are in 2 smaller hemizygous regions and are unlinked. So they're on different chromosomes. But in both cases, the genomic regions around the genes are associated with repetitive DNA. So whether the genes are in non-recombining regions, linked or unlinked, buried in repetitive regions, this genetic architecture affects how easy it is to find those causal genes for apomixis, because recombination has to occur to be able to map those genes. So why study apomixis in the Rosaceae? So this is a convergent trait. it pops up and disappears in many lineages across flowering plants. And Rosaceae happens to include a number of specialty crops that could benefit from being apomictic in a few different ways. And aside from the economic benefits, studying apomixis and Rosaceae makes sense if we want to understand the genetic basis and the evolution of the trait, because it ranks third for known gametophytic apomix. So this figure on the left is just a phylogeny of all the flowering plant orders, and next to the phylogeny, these columns are indicating the accounts of apomixis. So you don't necessarily need to know what each of the 5 columns means, just know that the darker the columns and the higher the numbers, the more frequent apomixis has been seen in that order. So if we're going to go about identifying the genetic basis in rosaceous genera and other lineages, that will provide us with more tools for plant breeding and inform us on the circumstances leading to apomixis to emergence and decay in flowering plants. And so the field is kind of moving now towards using comparative genomics to help us identify these regions, which is great because we're essentially taking advantage of nature's recombination and mutation rate over many generations, which you kind of need if you're looking for genes in recombination suppressed regions. But to do comparative omics, to find the genetic basis of apomixis in all of these different lineages, you have to phenotype for apomixis, and that's not an easy trait to phenotype. It's because you can't see it with the naked eye. You have to look at either microscopy, a development of the ovule, which is low throughput. You can use flow cytometry, which is a lot higher throughput, but it's kind of an art and it requires specialized equipment that not a lot of people might know how to use. And finally, the third classical way to do it is by sequencing both the mother and the child genotype and comparing their DNA sequences. So that used to be expensive, now it's getting cheaper, and most molecular labs are well equipped for DNA extraction and analysis. So that's how our lab is identifying apomix. And we're obtaining our diverse apple genotypes from the USDA germplasm collection in Geneva, New York, and where thousands of genotypes from all over the world are maintained. So I was there last spring for 5 weeks getting samples for my project, and in the process, also managed to get my little Saturn Aura stuck in the orchard the first week at 5 p.m. on a Friday. But I was rescued thanks to the excellent farm staff there. All right. So this is what our wet lab pipeline looks like for screening apomix. So I'll identify germplasm individuals that I'm interested in based on literature searches and documentations of apomixis previously. Then our collaborator will send us open pollinated fruit and seed, and then we take only the embryo, we place it in 96 well plates with 1 embryo per well, and then we create 96-plex sequencing libraries with kits made by Twist Bioscience, sequence these at low depth to save on costs, and then sequence the mother genotype as well to a higher depth. So 2 fantastic undergraduates in particular helped with the pilot study, Lauren and Trinity. Okay, so for the analysis, it's pretty basic. We just process mom and embryo sequence data in parallel. We demultiplex it, pre-process it with FAST-P, align it to the Honeycrisp genome for our reference, and then I actually use 2 different variant callers to call variants, and then unite the calls with GATK concordance function. And the reason I do that is because it really just eliminates that error rate in the calls. And so what we're doing really is just looking site by site with mom and embryo and seeing whether the genotypes at that location match or not. So here is what the data looks like. So this is for 16 apple genotypes. Each triangle is a single embryo and they're colored by mom, and they're also labeled at the top of their little respective plots. So this actually isn't a PCA, it's actually a bit simpler. On the x-axis, I have % of total sites that matched mom when she was called as heterozygous, and on the y-axis, homozygous. And the reason I did this was to give me an idea of what type of fertilization event may have happened to produce this certain embryo in question, so like a selfing or an outcrossing event. So for example, if an embryo was produced by selfing, it would have near 100% homozygous calls, but the similarity along the x-axis would vary because of segregation of those loci. So for this experiment, we had 2 sexual controls, this Malus spectabilis and Malus asiatus accession, and 2 apomictic controls, this Malus tupensis and Malus sargentii. So in the sexuals, can you guys see my cursor okay? Oh, right here? Yeah.
Yep.
So in the sexuals, excellent. So in the sexuals, you can kind of see like a cloud of similarities. So each embryo does deviate pretty significantly in terms of similarities relative to mom. But in the apomix, you can see the vast majority of the embryos here are clustering in this top right corner near 100% similarities for both types of calls. So that's excellent. That means our controls are working. But you also have these other embryos that are kind of scattered here and there. And remember, apple is facultative. So it is still capable of sexual reproduction. So we weren't necessarily surprised to see that. I'm trying to make my cursor go back and it's not cooperating. Ah, here we go. Okay, cool. Okay, so using this screen, we actually did identify 2 new apomix, which was great. The succession of Malus baccata, pretty much every single one of her embryos are up there in that corner, and the succession of Malus torringoides. So I've circled the embryos that we'd consider apomictic, and Malus torringoides only has a couple out of like the 20 or so that actually were produced apomictically, while Malus baccata there has all of them. So this is where we would say, these 2 individuals differ in their penetrance of the trait. So aside from these genotypes, all the other genotypes we'd classify as sexual since they showed substantial differences relative to male. Okay, so for the rest of my talk, I want to talk about the progress we've made in understanding the genetic basis of apomixis in apple. So recently I assembled 3 chromosome scaled genomes, including our 2 apomictic controls, Malus hupensis and Malus argentii for our screen. They're both triploid apomix and another genotype that's very likely misidentified in the germplasm database. It's labeled as Malus hupensis, but that is a species that is almost by definition triploid and apomictic, and this one is not. So this is a sexual that I think is Malus holliana. So never believe what is labeled in the germplasm databases is the lesson here. So assembled these genomes. For context, here's a PCA of some 263,000 SNPs, illustrating the relatedness among all these genotypes that I used to screen, and some I pulled from NCBI's Sequence Read Archive too. So here's the 3 individuals I assembled genomes for. They're all pretty closely related, and all of the individuals in this left side here have an Eastern or Central Asian origin. And for context as well, the cultivated apple is kind of over here. So that's Malus domestica. Okay. So we always have to kind of give some boring metrics for each assembly. So here's some contiguity and BUSCO information for the 2 haplotypes for my sexual diploid that I assembled. Each bar represents the number of contigs that make up a particular chromosome. So if it's one solid color, it's a single contig making up that chromosome. And if the color changes, you have a new contig. So these 2 haplotypes are pretty darn contiguous. We're missing a few telomeres, but honestly, it's not really a huge concern for my purposes. And you might also notice too, the BUSCOs look good, but there's a lot of duplicates. And that's because apple is a paleopolyploid that has retained a lot of its duplicates. So this circles plot is just showing the remnants of that ancient polyploid event by illustrating those syntenic relationships between its 17 different chromosomes. Okay, so this is the first apomix genome. So it's triploid, 17 times 3 is 51, and there are indeed 51 chromosomes. So I just broke them into 3 groups, honestly, just for visualization purposes. But the BUSCOs look good for here as well. And the contiguity is a little less, but I mean, this is a polyploid. So there were more paths in the assembly graph, so not to be unexpected. This is the other apomix, looks pretty good. So obviously, we're all fascinated by polyploid evolution here. So one of the things I like to do when I first assemble a genome is see if there's any sort of subgenome clustering in the chromosomes, or if there's like a distinct set of chromosomes that seem to have K-mer signatures associated with them. So I'm defining a subgenome here as having repetitive sequences that cluster these certain chromosome groups. And that assumption there is that those that have these fossil TEs, that shared fossil TEs, may have come from a similar ancestor. So I did some hierarchical K-mer clustering with Camille's pipeline, the ONO pipeline. And yeah, you just K-merize the genomes and the separate chromosomes, and you look to see if there are any patterns in the data. So, surprise, there weren't. So there weren't any clear clusters of complete chromosome sets in either triploid. But you could still see some clustering for certain chromosomes and haplotypes. So this is a heat map of the types and abundances of K-mers by chromosome for the 51 chromosomes in the first triploid, Malus hupensis, apomix number 1. So there are 671 different K-mers in this plot. Their abundances are shown according to the heat color. And on the x-axis up here, this is the dendrogram showing the clustering of the different chimeras. So the closer they are, the closer the sequence is. And the y-axis is showing how the chromosomes cluster according to the algorithm and the abundances and types of chimeras. So I've kind of made some little subgroups here based on I, and none of these contain a full set of 17. And in fact, some of them contain 2 copies of the same chromosome group. So we can look at our other apomix, which is a lot cleaner, but still no clean groups of 17 here. And so the question, of course, here is why? Why is this happening? And well, what's a species? So apples regularly hybridize between species all the time, and it seems like the chromosomes are regularly exchanging with each other, segregating across generations. And so that's probably preventing us from seeing clear subgenome groupings. And honestly, it makes a lot of sense in terms of what we see with other apomictic species. They often form giant complexes with where the species lines are blurred. So seems to be the case in apple as well. Okie dokie. So let's talk a little bit about the structural variation I'm seeing. Generally speaking, most Malus genomes are pretty synthetic, but I'm already finding that the apomix shows some major structural variation relative to the sexual genomes. So this is a dot plot for one group of the 17 chromosomes from our apomictic Malus hupensis here on the left and Malus sargentii on the right, and they are being compared against 17 chromosomes, so one haplotype of sexual Malus domestica. And yes, I chose this one haplotype for these triploids to show you, and you'll find out why in a second. So in both species, you can see that there's a breakdown of the synteny here and there. So that's represented where these nice clean diagonal lines kind of break and flip. So the thing I was looking for here is if apomixis has a single origin in apple, we should be able to look for genetic variation that's unique to both apomix that's not present in any of the other haplotypes from sexual species. So I kind of had this pattern in mind. And with this pattern in mind, the most obvious thing that caught my eye is that both apomix show a substantial breakdown of synteny in a really large region of one of their chromosome 5 haplotypes. So if we zoom in, we can really see how this region is rife with inversions and duplications in places with no synteny at all. So I actually wasn't the only one in our lab that was super excited to see this. Our lab also studies sex chromosomes, and this is the kind of pattern you would see in a typical hemizygous recombination suppressed sex-determining region of a sex chromosome. So that's a huge ongoing project as well. And part of my pitch when I was, applying for this NSF proposal was that, we're starting to see these loci look a little bit like these SDRs in sex chromosomes. And so if I join Alex's lab, I can learn a lot of techniques that might help me find them in apomix. Okay, so we can take a finer look at this region. So this is a plot generated with deep space for nucleotide synteny, and that's important because I don't actually have annotations for these genomes quite yet. But the 2 sexual haplotypes are up there on the top, Malus domestica haplotype A, and one of the haplotypes of that Malus fake hupensis that I assembled. So they're kind of our reference, and I intentionally alternated the haplotypes of our apomix here so that we could see the direct synteny between those 2 weird chromosome 5s in the apomix, the ones with those structural rearrangements. So locations highlighted in pink are indicating inversions, and so the region of interest here is right here. So these 2 chromosomes, they have some synteny, but it's kind of like you look at it and you're like, where do I start with this? It's a mess. There's inversions relative to some of these sexual haplotypes. And even between the 2 apomix, there's a lot of structural rearrangements. So something very interesting here is going on. So given that the loci in these apomix, though, looked like non-recombining hemizygous sex-determining regions, I decided to give a certain analysis a shot, one that my friend and colleague in the Harcus lab does when she is trying to look for sex chromosomes in new genomes that she assembles. So this is just a shared chimer analysis. Basically, it involves chimerizing DNA sequences and then finding chimers in groups of individuals that might be unique relative to other individuals. So when Sarah does this, she looks at unique chamers to groups of males and females. But in my context, I'm asking, what chamers do the apomix share that aren't in any of the sexual species? And this is based on, this has an assumption though. This is based, again, that apomixis has a single origin in Malus and that the genes conferring the trait are probably the same. Because if there were multiple origins and you had different genes, the resulting signal of shared chamers would either be like just noise or just downright disappear. So for this analysis, I used 5 apomix and 12 sexual species to chamerize and look at. And as a reminder, here's our PCA again, showing relatedness. This is where the 5 apomix fall on the PCA, and the sexuals are kind of dispersed all over the place, so I didn't label those, but I did list a few of them out here. And most of these we screened. But the basic steps for this pipeline were you count chamers in all of the individuals, and I found the ones that the 5 apomix shared, then compared those chamers to the 12 sexual individuals using Merrill's difference function. And then I simply used BWA-MEM to map them back to our apomictic genome assemblies to see where they lie and visualize the results in R. So for effect, I'm going to show you where the chamers unique to the apomix mapped on the chromosomes in descending order. And because we have 51 chromosomes, I kind of have to show it to you in 3 different pictures. So here are the 3 different haplotypes for our 2 apomictic genome assemblies. And this is groups, chromosome groups 12 through 17. So there's 3 of group 12, 3 of group 13, etc. So we have Malus tupensis on the left, Malus sargentii on the right. The density, if it had mapped here, would be in green and brown, but it's pretty much just a flat line because the chamers aren't mapping to any of these chromosomes. So this is chromosome groups 6 through 11, and it's kind of the same deal. You do see a couple of mysterious little peaks here and there, but I think those might just be noise. They're not really mapping in necessarily similar places. Okay, and so finally, this is chromosome groups 1 through 5. And there they are. Pretty much every single one is there in that weird, large hemizygous section of that haplotype of chromosome 5. So this was pretty fascinating to see. It means there probably is a shared origin for apomixis in these 5 apomix, because again, the signal probably would have disappeared if they had not had a shared origin. And they're mapping in similar places too. So this is where we would hunt for those genes. And if we go back to our synteny plot too, we can look at where those peaks are. They're here. And I'm especially interested in the regions of synteny that connect the red regions and are in the same orientation when comparing the 2 apomictic chromosome 5 haplotypes and show no synteny between the other haplotypes. So these might represent blocks of duplicated genes or unique sequence that may contain cis elements affecting the expression of some genes. At this point, we just don't know. So I'm working on getting some more data to find out. So in summary, our low-pass whole genome sequencing is an effective method to identify clonal progeny, but it is kind of expensive still. And I didn't really talk about this in my talk, but I am working to make this more cost-effective using custom probe design, because I really only need like so many sites to be able to confidently say that is a clonally produced embryo. And then the preliminary analyses I've done so far with the genomes I have assembled indicate that hemizygous region is associated with apomixis on chromosome 5. And the Kamer signal suggests that these sequences may be conserved in those 5 apomix with a single origin. But really, we need more genomes and annotations to start getting down to those candidate gene levels. Cool. So that's it. Lots of people to thank. And I'll take questions now if anybody has any.
All right. Well, thank you, Charity, for a wonderful talk. As always, people can feel free to raise their hand or put their question in the chat. And I'm going to get a question in real quick while we're waiting on other folks.
Sure.
That was really, really cool. So I was just thinking about the Kamer analyses. How old do you think those, just think of the subgenomes as well as some of these other regions, how old do you think these are? And do you think you're getting enough sort of repetitive element accumulation to distinguish those putative subgenomes in the first place?
So you're talking about trying to actually see different subgenomes, right? Like the groups of the
Yeah,
17?
for example, yeah, for the back in the middle of the talk, yeah.
I mean, it's hard to say how much is needed, I think, really. I mean, 671 very repetitive Kamers, I think, would probably be enough to at least see a strong signal. But if it's super weak, I also tried subphaser, which didn't
Right.
do it. Like just crashed. It's just
Yeah.
crashing over and over again.
I was just curious though if there was enough, difference, if there's enough divergence, I guess, in the first place to see to distinguish them. Is what I was wondering about. Yeah.
Yeah.
But
I
anyway,
mean, yeah. I mean, I can see some groups, right? But
right. Yeah.
I kind of doubt that they are like preventing segregation or recombination. I think they're all I think it's a free for all.
Right. Cool. All right. Any other questions or comments for Charity or for Christoph's talk? I know we're sort of running over time. I think we're leaving. Oh, Christoph has a question. All right.
Yeah, go for it.
Yeah. As an ecologist, so these what I didn't really get, probably because I was confused. These are different species or just let's say cultivars or
Yeah, the genomes that I assembled, is that those ones, or just
Mhm.
all of my talk was based on just the genus Malus. So I had different species in there. I had
Mhm.
cultivated
Yeah.
accessions, like old ornamental cultivars that you just use in landscaping. But
So
they
this means
all.
that
Oh, go ahead.
yeah, this means that there is some evolutionary sense in the evolution of the apomixis apparently. And my question would be, do you have any idea, maybe in terms of distribution or whatever, why in some lineages or some species has evolved the apomixis, like this Red Queen hypothesis and these kind of things? Is this something you also investigated in with your data, or at least thought about it? Because that's something which
Yeah.
always fascinates me. So.
Yeah, I've definitely thought about it. It's like the population genomics and the apomixis effects on those are less of a focus for my project. But I think apomixis is beneficial in certain contexts, of course. So if you have a set of genetics that happens to have, the right cards for a certain environment, it does make sense for you to just clonally reproduce and spread your genetics everywhere. But I also think too, it does benefit organisms that may hybridize and may not, like odd numbered polyploids that may not reproduce sexually very easily. So that's kind of a fallback on, now I will reproduce no matter what. So those were kind of my thoughts. But I haven't really studied apomixis and its effect on apple evolution as a whole.
And maybe one quick question: is this used in cultivars in breeding? So are the people breeding lineages that are particularly apomictic to avoid stress with honeybees, or depending on honeybees or frost?
No, that's a great question. People, for a plant breeder, it's actually not super advantageous to use apomixis in like developing new cultivars because you're sort of selecting against being able to recombine the genetics, right? It's more the utility comes from when you already have a really great genotype and you want to preserve that one's genetics. Does that make sense?
That makes sense. Thank you.
Okay.
Thank you.
Yeah.
It was a great talk, by the way. Wonderful.
Thank you.
All right. One last quick question here from Andre in the chat. It says, thank you for a very nice talk. Perhaps I just missed, how many samples have you used per species?
Oh boy. Most of those were just single accessions right now, but when I start to get my screen up and running where it's a lot cheaper, I do have like some 50 to 70 genotypes I'd like to screen that I collected from last spring when I was there. And so I tried to target at least a few of the a few accessions of the same species. But I'm also finding too that a lot of them are misidentified. And even though it says like this is a Malus hupensis, it's not. It's a Malus holliana. Or I also found a hybrid too that was supposed to be an apomictic accession, and it exclusively reproduces by sex, and its fruit is all wrong compared to what it should be. So you always have to take that into consideration too. But I am trying to have some replication with some of these as well. But sometimes they are not always as they appear.
All right. Well, thank you. That was really exciting. I might send you an email about a couple other questions about the
For
sex
sure.
determining regions and everything else. But that was really fascinating. And thank you to both Christoph and to Charity for these two great talks today. And thanks to everyone for coming out. We'll meet again on the first Wednesday of March. And our speakers then will be Maya Wilson-Brown and Nomar Wamanal. And I really look forward to hearing from both of them and hearing about their research. And keep an eye out for the email as usual, and we'll see folks in March. All right. Well, thank you.
Thanks, Mike.